Have you ever wondered why you're taller than when you were younger? Have you ever asked yourself why your bones heal after you break them? Well, that is because your cells, which are the functional and structural units of your body, are completing the cell cycle that is growing and replicating. The cell cycle is the life cycle of a cell. It is divided into interphase and mitosis. During interphase, the cell is growing and replicating its genetic material. During mitosis, a parent cell splits into two identical daughter cells. This type of cellular division takes place in your body's cells and is used to increase your number of cells or to replace dead or damaged cells. The first stage of interphase is called G1. Cellular content, excluding the chromosomes, are duplicated. At this point, a human body cell would have 23 pairs of chromosomes, with one chromatid each. Before the end of this phase, there is a checkpoint in which the cell checks that nutrients are sufficient, that growth factors are present, that the cell size is adequate, and that the DNA is not damaged. Some cells enter the G0 phase, which is a state of cell cycle arrest. During the second stage, S for synthesis, DNA is replicated and chromatids are duplicated. The centrosome, made of two centrioles, is also duplicated. At this time, a human body cell would have 23 pairs of chromosomes, with two sister chromatids each. In the third stage, G2, the cell prepares for mitosis and checks for possible errors. Prior to the end of this phase, there is a checkpoint in which the cell checks that chromosome replication is complete and that the genetic material is not damaged. When the cell is ready to divide, it enters the mitotic phase of the cell cycle. Mitosis is divided into prophase, prometaphase, metaphase, anaphase, and telophase. During the first stage, prophase, chromatin fibers condense into chromosomes. In a human body cell, the result is 46 chromosomes, with two sister chromatids each. First, the nuclear membrane and the nucleus disintegrate. The centrosomes separate and move to opposite poles of the cell, and the mitotic spindle apparatus emerges. Between prophase and metaphase, prometaphase takes place, in which kinetochores appear at the centromeres of the chromosomes and attach to the spindles. In metaphase, chromosomes are aligned at the metaphase plate or equatorial plate, perpendicular to the filaments of the mitotic spindle, which is completely developed. During anaphase, the sister chromatids of each chromosome separate as they are dragged by the spindles. Each chromatid travels to one end of the cell. In the last stage, telophase, the daughter chromosomes migrate to the end of their corresponding cells. The spindle apparatus disappears and chromosomes unwind into chromatin. Thus, two nuclei are formed, surrounded by enveloping material. After the different mitotic stages, the final separation takes place during cytokinesis. In animal cells, a cleavage furrow separates the two identical daughter cells. Each daughter cell has a nucleus containing 46 chromosomes, with one chromatid each, and of course, distributed organelles.